Hello, my name is Anna Cehovin, and I'm a postdoc in the lab of Professor Christoph Tang at the Sir William Down School of Pathology. And today I will tell you about mobile antimicrobial resistance in Neisseria gonorrhoeae. So Neisseria gonorrhoeae has developed resistance to all antimicrobials used in its treatment, and the WHO recommends dual therapy for the treatment of gonococcal disease, so the use of cefixime or ceftriaxone in combination with azithromycin. However, the CDC already recommends monotherapy, so the use of only cefixime or ceftriaxone, and this is to prevent the rising resistance against azithromycin. There are many resistant determinants in Neisseria gonorrhoeae, which can be either chromosomally encoded or plasmid encoded. And in terms of plasmids, Neisseria gonorrhoeae harbors three plasmids: the conjugative, beta-lactamase, and cryptic plasmid. The conjugative plasmid is the biggest plasmid, and it can carry the tetracycline-resistant cassette, but it can also be markerless, so without any antimicrobial-resistant cassette. The beta-lactamase plasmid always carries the beta-lactamase cassette, which confers resistance to penicillin, and the cryptic plasmid is the smallest plasmid and does not carry any antimicrobial resistance cassette, and it also is not associated with any phenotype. So when we were looking at the plasmid prevalence in global gonococcal populations, we found that there are big discrepancies between countries. So on these graphs, each dot represents a country, and the size of the dot represents the number of isolates that we analyzed from that country. On the x-axis, there's gross domestic product, or GDP, and on the y-axis, there is prevalence of the conjugative plasmid with TETM, the beta-lactamase plasmid, or the markerless conjugative plasmid, which does not have any antimicrobial resistance. You can see that as the GDP of a country decreases, the prevalence of antimicrobial resistant plasmids decreases. And this is true for conjugative plasmid with the tetracycline resistant cassette and the beta-lactamase plasmid. But we did not see the same trend with the markerless conjugative plasmid. So it is clear that there is a high frequency of AMR plasmids in low and middle income countries. And this is probably due to the syndromic management employed in these countries for gonococcal disease. So we asked the question, what is the consequence of such a high prevalence of AMR plasmid in gonococcal population? And how would this plasmid be maintained in a gonococcal population? So to be able to answer these questions, we first set off to characterize plasmids in Neisseria gonorrhoeae. So we first wanted to analyze phylogenetically our data set, which consisted of over 3,700 gonococcal isolates from 56 countries that were collected over decades. And we determined the phylogenetic relationships using the core genome MLST scheme, which clusters the isolates according to over 1,600 core genome loci. We employed the scheme CGC 400, which clustered the isolates with less than 400 allelic differences. And on this phylogenetic tree here that you can see, each isolate is represented by a circle, and each circle is colored according to the lineage that this isolate belongs to. We then wanted to determine the global distribution of plasmids in these lineages. And we first defined and curated the plasmid genes. So 50 genes for conjugative plasmids, 8 genes for the beta-lactamase plasmid, and 9 genes for the cryptic plasmid. And all of these are stored in the plasmid scheme on PubMLST. This is the same phylogenetic tree as you saw before, except that now the isolates are colored according to the plasmid types that they harbor. So isolates, for example, isolates in lineages 16, 3, and 18 are colored in blue because they carry only cryptic plasmid. However, isolates in lineages 21, 109, and 33, for example, are colored orange because they carry all three plasmids. An interesting one is also lineage 15, which is colored here in light orange because it doesn't carry any plasmids. So it is quite clear that plasmid type distribution is associated with specific lineages. We then went on to characterize the conjugative plasmid. And in the literature, only 3 types of this plasmid are distinguished: the markerless, and the Dutch and American according to the TETM allele that the conjugative plasmid has. However, when we aligned long-read sequences of this plasmid, we found that many more variants of this plasmid exist, and these variants differ in the mating performation region and in genetic load region. So to be able to distinguish these variants, we employed a clustering algorithm which clustered plasmids according to 5 allelic differences. And this gave us 26 clusters of the conjugative plasmid. And we then constructed a phylogenetic tree based on all the loci found on the conjugative plasmid. And with this phylogenetic tree, we were able to distinguish 7 variants of the conjugative plasmid. And when we aligned the genetic load and the mating performation region of these 7 variants, we found that these variants differ in antimicrobial resistance, so the TETM cassette, in TRBK and TRBL genes, which are involved in the plasmid acquisition, and also in epsilon zeta toxin-antitoxin systems, which are involved in plasmid stability. We then wanted to know how these variants are distributed in the gonococcal population. And most variants are found across the gonococcal population. However, variants 2, 4, and 7 are found associated with particular lineages. And currently, work is underway in our lab to try and understand which factors or either or the host microbe or the plasmid are involved in maintaining these plasmids in these lineages. We also wanted to characterize the beta-lactamase plasmid, and you can see from this alignment that many variants of the beta-lactamase plasmid exist. However, the repeat sequences on this plasmid prevented the correct assembly of the short sequences. So we could not employ the same plasmid typing schemes that we developed for the conjugative plasmid. So instead, we determined the distribution of this plasmid according to the TEM allele that is found on the beta-lactamase plasmid. So the most prevalent TEM allele on the beta-lactamase plasmid is TEM1. And it's here in dark blue, and you can see that it's uniformly distributed across the gonococcal population. And this TEM1 beta-lactamase needs only 2 amino acid substitutions to become an ESBL, or extended spectrum beta-lactamase. And an extended spectrum beta-lactamase would be resistant to cephalosporin and cefixime that are currently used in gonococcal for the treatment of gonococcal disease. We also, however, found that the TEM135 allele of the beta-lactamase, which already carries one of these substitutions, is found in specific lineages 20, 25, and 109. So why is this important? It is important because our data shows that beta-lactamase plasmid is found more often with the conjugative plasmid than without. And this is because the beta-lactamase plasmid is non-conjugative and it needs the conjugative plasmid to be able to be mobilized from one bacterium to another. So gonococcal populations that contain a stable variant of conjugative plasmid and a high prevalence of conjugative plasmids will be better at spreading the beta-lactamase plasmid. And if this beta-lactamase plasmid contains an ESBL, for example, this would mean that ESBL could actually rapidly spread through a gonococcal population, and it can lead to untreatable gonorrhea. So we are now trying to understand why certain populations have a high prevalence of plasmids and why certain populations have low prevalence of plasmids. And as part of this, we're trying to determine whether restriction modification systems are associated with either high or low prevalence of plasmids. So restriction modification systems are defense systems that can restrict the invading DNA, including plasmids. And gonococci contain 3 types of RMSs, which we are now curating, and they are now found in this RMS scheme in Neisseria gonorrhoeae on PubMLST. So in total, there are 33 loci of RMSs, and I am now trying to determine whether there are associations with lineages with either high or low plasmid prevalence. I'm also trying to detect any specific signatures on genomes and plasmid to try and understand which of these RMSs are involved in plasmid mobility. Okay, I will finish here. I would like to give my thanks to Professor Kisop Tang and all of his group. Massive thanks also to our collaborators, Professor Martin Maiden, Dr. Odile Harrison, and Dr. Keith Jolly for curating the PubMLST. And also big thanks to our collaborator in Kenya, Dr. Edward Sanders. And thank you for listening. And I would be happy to answer any questions. Thank you.